And so one of the most common ways that's used to separate gram-positive coccus from each other, and when you look at your microscope and you see this underneath the microscope, it's probably going to be one of two things that are causing problems in your patient. It's probably going to be either a staph or a strep. Not guaranteed, but more than likely it's what it's going to be. Especially if it's something on the skin or something from the throat area. It's going to be one of those two guys. So anyway, you'd see this under a gram stain. They're both going to be purple. How are you going to tell them apart? Well, you can't put staphs on an MSA, or streps on an MSA plate. They'll die. The salt will dehydrate them. So if you try that and you I'm going to put them on an MSA plate and see what type of staph it is. You put it on there and it's actually a strep. They're going to die. You won't be able to see them at all. That's really important to make sure that you actually get the diagnosis if it's a strep or a staph correct, so you don't put them on the incorrect auger and they accidentally die. You have to start all over again. So anyway, this is used to differentiate staph species from streps, those two guys. So how do you tell them apart? They're both gram-positive coccuses. Well, one of them is going to produce an enzyme called catalase, and the other one is not. Staphylococcus species are all catalase producers. They can all produce the enzyme catalase, and Streptococcus species cannot. They cannot do this. So what is catalase and what is it used for? Well, inside of your body, your red blood cells use lots and lots and lots of hydrogen peroxide to help destroy bacteria inside the immune system, things like peroxisomes and things like that. And also bacteria that use oxygen, any bacteria that uses oxygen, if it comes in contact with oxygen in its metabolism, will break down that oxygen into toxic byproducts that'll build up inside of its cell and cause damaging DNA effects and things like that. And one of those products is hydrogen peroxide, also found in those peroxisomes. So both of those things, both of those sources of peroxides in the body. So anyway, if you use oxygen, you're going to make those toxic byproducts, and if you get inside the body or you're pathogenic, you have to deal with that possibly as well, getting attacked by the hydrogen peroxide. Well, anyway, so if you produce hydrogen peroxide, it's very toxic. Hydrogen peroxide comes in contact with water inside of the body, and it breaks apart into things like superoxygen and hydroxide free radicals and things like that, which are very, very, very dangerous. Hydroxide free radicals will act like magnets, and they rip apart DNA and things trying to get to one another, rip apart cell membranes, cell walls, and just tear bacteria apart from the inside out. And those other superoxide products and things like that are also very dangerous to bacterial's DNA. Well, anyway, if you use oxygen, you will produce those toxic byproducts. There is no ifs, ands, or buts about it. So you have to have a way to get rid of those toxic byproducts, or else they're going to build up inside of your cell and you're going to die. So how do you get rid of them? This enzyme right here, catalase. If you can produce the enzyme catalase, you will be able to break down that hydrogen peroxide that you produce through using oxygen. If you can't produce the enzyme catalase, the hydrogen peroxide that you come in contact with will kill you. It will break apart down and you will die. So if you produce the enzyme catalase, if you're a Staphylococcus species that lives on the skin, these guys are aerobes, they live on the skin, they use oxygen, so they have to be able to break down those toxic oxygen byproducts. So they come in contact with hydrogen peroxide, and the catalase enzyme will break down hydrogen peroxide into water and oxygen, which is completely harmless. Both of those things are totally harmless to the bacteria. They don't mind that at all. So if you cannot break down the hydrogen peroxide, if you're a Streptococcus species, you come in contact with hydrogen peroxide, you don't have any catalase enzymes to break it down. Streptococcus species are facultative anaerobes. They don't, aerotolerant anaerobes, they don't use oxygen. They cannot use oxygen. So hydrogen peroxide will kill them. They don't have any way to break it down. They don't produce hydrogen peroxide through their metabolism byproducts, so they don't have any way to get rid of it. So if they come in contact with it, it will break apart inside of their bodies into free radicals and things and kill them. So Streptococcus species will die in the presence of hydrogen peroxide. Staphylococcus species can survive in the presence of hydrogen peroxide for a little while until they run out of catalase. So how do you tell them apart, and how does this test work? So the background: staphs can survive in hydrogen peroxide. Streptococcus species cannot. So you're going to take a big sample from your patient, or from your petri dish, excuse me, or your agar or whatever it is. You can see they've got agar slants down here. You take a big sample from it, and you put it on a slide that's going to contain some liquid hydrogen peroxide. You can see that here. Well, if the organism produces the enzyme catalase, it is a catalase-producing organism, usually is some sort of staph species, you will instantaneously see massive bubbles. The hydrogen peroxide, the enzyme catalase, is breaking apart that hydrogen peroxide into the oxygen, which is being bubbling up inside of that water, making oxygen bubbles. So this is the process of the hydrogen peroxide being broken apart into oxygen and water. The bacteria is doing that. So let's pretend you have a catalase negative organism, catalase non-producing organism. You scoop a big scoop of them up on your loop or on a swab or something, and you put them in contact with hydrogen peroxide, and nothing. They cannot break down hydrogen peroxide. They don't produce catalase that will break down hydrogen peroxide, so no bubbles at all. This organism is dying. It is dead right now. This guy can survive as long as they're producing bubbles. You can do this test with loops on a slide. You can do it in a petri or in a tube with hydrogen peroxide, liquid hydrogen peroxide, and little sticks and things like that, and watch for bubbles. There's multiple different ways to do this, but it all works the same. You're looking for the presence of those bubbles. So if you have bubbles, you know this organism is able to produce the enzyme catalase, which means that it is a Staphylococcus species. So then you would proceed to an MSA plate to tell apart which species of Staphylococcus you have. They can all produce the enzyme catalase. If you have a catalase negative organism, a non-producing catalase organism, it's probably some sort of strep. If you put that on an MSA plate, it will die. So you'll go to a blood plate. And then look at the types of hemolysis to tell it apart what it does. And this is a little video right here if you want to watch how that does. So Catalase Test tells apart species of staph from species of strep, so you know which step to take next to put them on agar to tell them apart. And it does so by looking for the presence of catalase-producing bubbles. The organism produces the enzyme catalase or not, and that's what the Catalase Test is used for. And our last test is the Oxidase test. And this is our catalase test for gram-negative organisms. This is going to be used to tell apart members of our Enterobacter Ace family from the other large family of gram-negatives that cause problems in humans, the Pseudomonads. So Pseudomonads are the only ones that are going to produce this enzyme oxidase, and this is the same thing for these guys. This is what they're going to use to break down those toxic byproducts of oxygen, cytochrome C. Cytochrome C oxidase is what this Pseudomonas organism is going to use to break down their toxic byproducts of oxygen. So anyway, scientists have figured out that this little compound here, TMPD, can be used to trick cytochrome C. You can detect the presence of cytochrome C by turning it purple. So the cytochrome C will accidentally donate some electrons to this chemical instead of getting rid of them like it should be doing in the bacteria. So when cytochrome C comes in contact with this chemical, this is the oxidase test reagent. You don't need to know that. That is totally fine. It turns purple. So cytochrome C will donate some electrons to this instead of donating them to the electron transport chain in the bacteria like it's supposed to, or excuse me, serving as the terminal electron transport receiver as it should. Well, anyway, long way to say this. You have two little slides. You can do slides that have this TMPD embedded on it. You can do a piece of paper with a little drop of TMPD on it, or a cotton swab that you put a little drop of TMPD on it. It's also called oxidase reagent. And then you take a little swab of your bacteria and you rub them on the little slide, or you rub them on the paper, and then you can rub the cotton swab on the bacteria as well and collect some. You let it sit for about 15 to 20 seconds, and then that TMPD will instantaneously turn purple if the organism is a Pseudomonas species. That's an organism that's able to produce this enzyme cytochrome C. So this organism is oxidase positive. These organisms down here that don't change color, or you can see here they stay the same, they are oxidase negative. They cannot produce the enzyme cytochrome C oxidase. They cannot do it. So this is how you tell apart the Pseudomonads from the Enterobacter Ace species. So you get a patient that has a gram-negative infection. You see that underneath the microscope. You grow it on a petri dish, and this is the first test you're going to do, just like a Catalase test, to tell apart your gram-negative coccuses. This is the first test you're going to do to tell apart your gram-negative rods. Is it an Enterobacter Ace species or a Pseudomonad species? If it's a Pseudomonad species, you're done. You know what to do. You can treat that. If it's an Enterobacter ace species, you have to do some more tests to figure out which one it is. So this is how you tell apart the two different broad families of our gram-negatives. This is essentially the gram-negative version of a Catalase test to break apart the staph and the streps. This breaks apart our Pseudomonads from our Enterobacter ace species. And you can see a little video here if you would like of someone talking through this as well again. So this is the two big tests here that are used to differentiate the two broad groups of our gram-negatives and our gram-positive bacteria. So if you have any questions over this stuff, guys, please feel free to email me. If not, have a good rest of the day.